Good morning.
Alright,
Can you
Assalamualaikum.
guys hear me?
I'm Javed.
Yes, sir.
Great. Thank you.
Assalamualaikum, sir.
Wa alaikum assalam. Looks like a nice big group over here.
I'm so sorry, I just got disconnected. The topic of today's seminar is a pain, drug, and addiction, like how electrophysiology helps build a connection. And the today's speaker is Dr. Arsalan Yusuf, and I am My apologies for unstable connection. So I was introducing Dr. Arsalan Yusuf. He secured his PhD in 2011 from Medical University, Vienna, Austria. And after receiving his PhD, he briefly joined Atatur Rahman School for Applied Biosciences, NUST as an assistant professor, but later moved to Sydney, Australia in 2012. Here he joined the lab of Professor McDonald Christie as postdoctoral researcher in University of Sydney. In 2019, he moved to University of Wollongong, Australia, where he joined hands with distinguished Professor David Adams' Works Group as a research fellow. His main research had always focused to understand molecular mechanisms of pain signaling from peripheral nervous system towards the brain. Over these years, Dr. Arsalan has worked with different G protein coupled receptors and their molecular regulation of membrane ion channels and second messenger signaling pathways. He has gained vast experience in cutting-edge tools like patch clamp electrophysiology, in vitro brain slice electrophysiology, and molecular biology. So over to you, Dr. Arsalan.
Okay. Thank you very much for the introduction, and thank you for this opportunity. I hope everybody can hear me well and clear. Just a heads up, if something goes wrong or if you think that my voice is not like being communicated properly, just pass me up. I'll share my screen, and then we can start from there.
Dr. Arsalan, we can hear you properly.
Okay. And let me know if you can see my shared screen now.
Yes, yes, we can see.
Okay, so if there are no further problems, I'll just start. So Like the host pointed out, I have been involved in research with pain research for the last more than 10 years or so. It involved my duration of my PhD and then, more than 10 years of my experience as a postdoctorate researcher and a senior research fellow. When I started my PhD in Vienna, my main interest was always, trying to understand and to study the membrane G protein coupled receptors and, their interaction with membrane ion channels. And, from time to time, it just stepped on to us. And then, I started becoming interested in, G protein coupled receptors that were basically involved in regulating pain pathways. And then, from Vienna, I joined Sydney, and then, the search and the hunt kept on going forward and forward. And then, I switched to different types of GPCRs, learned different techniques, but the main idea was always to actually investigate if, your body suffers any sort of injury, whether it's chronic, whether it's acute. Unfortunately, Allah maaf kare, whenever you get hit by a car or a road accident, or even like a finger prick or something, what actually happens? What's the main mechanism that is involved that allows the signal from the periphery, from the tip of your finger, that communicates all the way to the brain and gives you the warning signal that that thing actually hurts. So in this presentation, I'll go through some of the research that I've worked with Sydney University with my former lab supervisor, Professor McDonald Christie. I've joined University of Wollongong since like the last 1 year, but again, the focus has been on identifying the interaction between GPCRs and ion channels involved in pain. So yeah, let's start. When we look at the first slide, and an old cartoon character saying to you that pain is good, it actually is the truth, because if your body doesn't feel pain, if your brain system doesn't tells you that something is wrong, you would never be able to look after yourself. So pain itself is a good warning sign, telling you every time that something is wrong somewhere in your body. So it's a good defense system. It's a good warning system for your brain and for your healthy body. So what itself is pain do? It actually creates an alert system that something is wrong. Basically, whenever you hurt your body, or let's suppose you're playing, or like you're fixing tools, or you're mending your house or window, and you stab your hand with the hammer. The first actual response that goes straight to your brain within a fraction of a second, that ouch, that seriously hurts. This is actually caused by damage to the somatosensory system. That is pretty much spread throughout your whole peripheral body and communicating to the brain. And it's basically the activity of the CNA fibers, that are spread from the periphery. And, the information that goes straight from the peripheral sensory nervous system to the spinal cord, where it meets with the dorsal horn neurons. And then from there, it goes straight to the cortical regions of the brain. And there exactly, the brain actually communicates to the remaining part of the body, or to the warning system, or, takes the message straight back to the hand, and you see a withdrawal of the hand. But it took me like 2 minutes to go through this. Exactly, the response time is like so quick, within a fraction of a second. And that is basically, because of these myelinated CNA fibers that transfer the message so quickly. And whatever complicated things happens in between, we'll just take a small part of that chapter in this talk. So that's actually, something that can be termed as acute pain. Acute pain is something that, you hit yourself, you feel pain, your tissue just, after time, it gets recovered, it gets healed, and then, your pain goes away. what about chronic pain? Chronic pain is something really serious, and that actually happens. Sorry. So that actually happens when your body or any sort of tissue injury becomes so severe that it remains that injury. It's some sort of makes some plastic changes within your nervous system, the dorsal horn, or in your peripheral sensory system, that the body actually heals, but the pain signal still keeps on firing. And that is actually called chronic pain. That actually, your body still feels that pain, for an unlimited period of time, even, your body has completely healed after months, like, after, let's suppose after a severe car accident or something. And this chronic pain is actually so severe, it affects 20% of the world population. And, since USA is the biggest economy along with China at the moment, it costs US economy $600 billion every year. And, that's where our research, my research, is of great interest, that, when a person actually suffers from chronic pain, you need to have proper analgesics. You need to have proper medication. And that's, what we are actually interested in, to target novel medications, novel pharmacological drugs to actually effectively treat chronic pain. So yeah, but this actually leads to another serious problem. Not all of these medications have, good positive effects. Not all medications are effective analgesics, but most of them, which are effective analgesics, they can come with their side effects, like addiction, tolerance, dose limiting side effects. So yeah, my challenge, along with all the pain researchers in this field, has always been, starting from how the mechanism of pain can be well understood, and then trying to identify the molecular mechanisms and the molecular partners within the brain nervous system. Taking that, trying to identify novel analgesics, novel antagonists and agonist drug that can, block certain pathways and that can lead to relief of pain, but also to identify and to study the side effects, what actually goes wrong when an overdose of a drug happens, So yeah, there's like an ongoing quest. And when I said that, that it's actually, the talk is actually about building a connection between pain and, with drugs and, with the side effects. We'll just go through one part of, we'll just go through one of the most commonly known analgesics that have been known for centuries. So I'm sure everyone knows that opioids are the best analgesics by far to treat chronic and acute pain until now. And they have been, the best known opioid analgesics for centuries, even before, starting from 16th century. So my research, the last 7 years of my research in Sydney University was basically focused on, the G protein coupled receptor systems that were involved in opioid addiction and in opioid treatment of chronic pain. So yeah, that's the point where I'll focus on in this hour. So opioids, whether it's a blessing or a curse, like now, because we know a lot about the opioids and how different opioids can have different side effects and adverse effects. But it wasn't like a long time back, when opioids were actually treated as a wonder drug. In 19th century, morphine itself, which is, the first opioid that was actually discovered, it was actually given to children, when they had their swollen gums and when they had pain in their tooth, because it wasn't known that what their side effects are, but it was known definitely that this soothing syrup was a remarkable analgesic drug. And, morphine was actually used really commonly in the American Civil War to treat, war patients with, chronic pain and with serious injuries. And even until now in hospitals, if you, go through the hospital profiles where patients come with accidents, even in Pakistan, morphine and, opioids and steroids, they are commonly used to treat, to actually, relieve the patient of pain. But, when the time progressed from 19th century and when it became known that, there should be a way how morphine can be better designed to, so that, you can manufacture a better opioid drug with less side effects. And that actually, funny story, that actually led to the discovery of heroin. So heroin was actually, it actually came out because people actually wanted to find out a better cure than morphine, but they developed heroin, and which was actually even more stronger, more potent, and it was more addictive to the human body. And, people actually started getting to hit heroin like way more than 10 times or 100 times than they started getting addicted to morphine itself. So this history of, drug designing continued on, and, people, the chemists, they actually, later on, they designed methadone and fentanyl and oxycodone. So at the moment, there is a serious problem in the pharmaceutical industry. What different types of opioids are doing and how much of a disaster they have been playing in the world population worldwide. I have got 3 charts that I will discuss with you, which actually would tell what the status is of all these different opioids. So yeah, when we talk about opioids and about opioid side effects, these are the main top 5 side effects, and their main function that actually occurs. So opioids, definitely, they are wonderful analgesics, relieve pain abruptly and for a longer period of time. But when we talk about their side effects, respiratory depression is a very serious side effect that can occur with an opioid overdose, especially with heroin, with fentanyl, because fentanyl overdose is a serious problem in the United States, with oxycodone and morphine. Then, constipation, tolerance, and addiction. So tolerance and addiction is a serious problem with opioids, as you might all know. How actually people and patients, they become so much seriously addicted to these opioids. So I will actually discuss that in my coming slides as well. So here are some alarming figures. If we talk about USA, opioid overdose has been a serious problem. It's continuing even further up the chart in recent years. Fentanyl overdose has been a serious problem in USA, and the NIH, they are working extremely hard and, giving so much research grants to scientists over there and around the world to design better and safer analgesics and safer opioids. If we talk about Australia, in Australia, the opioid overdose death has been, climbing up and up because of the oxycodone crisis. But let's talk about Pakistan as well. I mean, it's this report isn't that recent, but in 2015, there were 2-3 reports that I could find that showed that how heroin is a serious, and opioid overdose is a serious problem, even in Pakistan. It was termed as an emerging crisis, if that was ever discussed, because, and the country found its place in the top 10 countries with the highest rate of opioid addiction. And this was actually an article in the South Asia Channel, where it was like clearly mentioned that, every day 700 people died from drug addiction, which was even greater than, the amount of people killed in terrorism at that time, which killed 39 people a day. So yeah, I mean, in Pakistan, it's really underrated how much serious the problem with opioid addiction is. Even until now, I don't think so. I mean, in the urban hospitals over there might definitely be reports of how many opioid addicted patients are coming. But it's an enormous challenge in a country like Pakistan, where the opioid crisis is, ongoing. So that was the scary part. Now let's just get a bit into the science. What actually happens in the brain whenever, somebody takes an opioid. So opioids actually, they specifically target this class of G protein coupled receptor, which is the mu opioid receptor. So whenever orally you take any opioid like morphine, it actually targets, the midbrain region, which is here, the brainstem. And, because the locus coeruleus, the LC region, this is, so this is the locus coeruleus region, is the basic region which is supplied with the mu opioid receptors, along with the VTA, the ventral tegmental area. So whenever you take an opioid, it actually acts on the mu opioid receptors in the brainstem. And what actually happens is it follows a chain reaction, in an upward trajectory. Yeah. So what happens is, from the locus coeruleus, the mu opioid receptors, they target the ventral tegmental area. And then when this area gets triggered, it sends signal to another region of the brain, the nucleus accumbens, which releases dopamine into the brain. And that neurotransmitter, dopamine, is basically, the main neurotransmitter that, is basically involves the reward and the pleasure and the excitement and the happiness. So, even under normal conditions, when you go for a run, you do an exercise and you feel really good about it, or, you take a delightful pizza or good food, you feel really good about it. Why? Because your brain is actually, producing a lot of dopamine. So there is a natural way how your brain can produce dopamine to reward your body and to make you feel good. And then, there is an oral way how you can actually take an opioid. And then, it actually, apart from being a good analgesic, it also triggers the dopamine reward system, which triggers your mood to make you feel good. So with this dopamine reward system, it also triggers the prefrontal cortex, and it goes back as a like a cyclic loop back and triggers back the VTA to produce more dopamine. And the locus coeruleus portion, which is this tiny portion over here, this is also the main region that is involved in in mediating tolerance and addictive profile. So what happens is when you stop taking an opioid, like the injury is healed and you stop taking an opioid, this feedback loop system, the reward from the prefrontal cortex, it actually keeps on triggering back the VTA and the locus coeruleus neurons again and again to get more and more opioid intake. And that actually, results in cravings and, side effects like diarrhea and respiratory failure. And now let's just look at the cellular mechanism, what actually happens at the cellular level. So whenever opioid targets the mu opioid receptor, what happens is you have ATP converting enzyme, which converts ATP to cyclic AMP. And then this cyclic AMP leads to increased noradrenaline production. And this noradrenaline is another G protein coupled receptor, and triggered by noradrenaline, which leads to excitement and flight response, flight or fight or flight responses. But when your body stops taking opioid, or when your body is becoming addicted to opioid, when you take more and more opioid, you will have more increased, double the amount of noradrenaline production. And that actually, keeps on, it makes like sort of like a connection and like a sort of a circuit that keeps on demanding more and more opioids, so that, they can increase more and more noradrenaline production, resulting in, the symptoms of withdrawal. Because, the receptor still is active, the receptor still is under the tolerant phase, but it's not getting the proper dose needed, which keeps on increasing and increasing. So now from here onwards, I'll go to the biophysical part of how, at the molecular level, the opioid receptors signaling actually happens. What actually is the key mechanism involved that leads from receptor desensitization and onwards. So a basic mu-opioid receptor is a G protein coupled receptor, like I said, with 7 transmembrane domains. And if in later classes, or even in your recent classes, if you've studied the G protein coupled receptor cycling, it is actually built up with 7 transmembrane domains with N and C termini. And then, it has got alpha and beta gamma subunits. So whenever any agonist binds to the receptor, the alpha and beta gamma subunit, they split up. And, these subunits can further activate further signaling mechanisms, which we will discuss as well. But what happens is that the C-terminal tail it just becomes exposed to different kinases in the cytoplasm. And most of these kinases, the most important kinase is the G protein receptor kinase, which comes and phosphorylate the C-terminal tail. And this just happens, very quickly within seconds. So as soon as the agonist binds to the receptor, you have the kinases coming and phosphorylating the receptor tail. And then, this leads to beta-arrestin protein binding, which actually halts the signal, the G protein mechanism straight away. So this cycle actually keeps on continuing until the receptor actually gets internalized into the cytoplasm, and then, it can recover back to the cell membrane again. And this is actually the key sequential terms, sequential processes that are involved in receptor desensitization. So whenever any opioid drug binds, it leads to, at higher doses, it leads to receptor desensitization, which means that you still have the drug bound to the receptor, and the receptor is still functioning, but its function starts to lower down with the passage of time, because the receptor itself is, it's becoming more and more desensitized. But, the surface resensitization on the membrane, it can happen over a period of time within like 10 to 15 minutes. And from the cytoplasm, from the intracellular domain, it can recover back in a matter of hours. Let's just not make it too complicated at this point. But the main take-home message is, sorry, you guys have to deal with this. The main take-home message is that the mu-opioid receptor activation, they inhibit calcium channels along the synapse to block the neurotransmitter release. And then, they also activate a class of potassium channels called G protein, inwardly rectified potassium channels, to induce membrane hyperpolarization. And, the signaling of mu-opioid receptor is very complicated. It's not just, one protein, one ion channel, or one protein, one signal. There is a plethora of different G protein dependent and independent signaling that happen at the same time. And depending on, which type of opioids are being used, for example, there is a difference in, the type of signaling that happens when you prefer morphine or over fentanyl, or different types of G protein signalings or G protein independent signalings are happening at the same time, which is quite complicated when we study them in later detail. So going further into the structure of the mu-opioid receptor, the C-terminal tail of the mu-opioid receptor has around 11 serine threonine residues, if you can see all the colored circles here. And most of these sites, they are like, they become phosphorylated by the kinases, like I mentioned. So when I started this project in 2012, if we can take the C-terminal tail into the next slide. When I started this project, the whole idea was to identify, because it was known that somehow the C-terminal tail of the mu-opioid receptor is somehow very important in regulating the mu-opioid receptor desensitization. So, for example, if these are HEC293 cells transfected with the mu-opioid receptor, and you can see that the green signal actually shows, the fluorescence by mu-opioid receptor using mu-opioid receptor antibody, that how actually it's signaling so brightly within the cytoplasm and on the cell membrane. And this is actually the wild type mu-opioid receptor with all the green colored serine threonine residues. So our collaborators in Germany, they designed different types of mutations in the C-terminal tail, which actually initiated this interesting approach. So one of their famous mutation was where they mutated all the 11 serine threonine residues to alanine in here. And then, they picked out some important residues, which they termed the 6-STA because it has 6 residues mutated, and the 3 STAs with 3 important residues. And they wanted to look at the receptor internalization. So the only thing was, you apply an opioid, for example, morphine or any other opioid like fentanyl, and look at receptor internalization. And you can see that in the wild type receptor transfected cell, you can see the green patch inside the cytosol. But in the 11-STA, upon treating them with morphine or fentanyl, you don't see any green patch or any internalization intracellularly. And same goes for the 6-STA. But somehow for the 3-STA, you still see internalizing. And the reason why this is happening is it was already known that these sites in the serine threonine residues, the STA and T region, is the basic region where beta-arrestin binds. And this is the region when it gets mutated to alanine, it actually halts receptor internalization. So that's what's happening here. The STA and T region here, when it gets mutated to alanine, you don't see any receptor internalization. Same goes over here in the 6-STA. But however, in the 3-STA, there was a bit of a tricky one because there is one site mutated out of the other ones, but you can now start to see internalization. So the whole idea was that to initiate this project and to actually understand how the surface desensitization, if it occurs on the membrane. And in order to do that, we had to first actually look at the phosphorylation by different types of opioids. So I'll just go through it very briefly. So Met-Enkephalin is actually, it's like a modification of the naturally occurring endogenous enkephalin that is produced in the brain, but it's a very strong opioid peptide, and morphine, as we all know. So the important part was to actually identify by using these site-specific antibodies what different types of sites are actually gets phosphorylated. So you can see with Met-Enkephalin, it actually phosphorylates pretty much all the sites colored very quickly within 20 seconds. But morphine, the phosphorylation is very weak. It doesn't phosphorylate most of the residues. It do phosphorylates the S375, but it's pretty weak as compared to what you see for the enkephalin. Yes, so to study the membrane properties, the electrophysiological properties, the idea was to get a good patch clamper, and that was me. To actually study the electrophysiological properties of these mutants and to see how the receptor desensitization can actually happen at the cell surface. So a little bit about patch clamping is you need like a massive, good, big microscope with everything, completely isolated from the ground. It has actually an air table that actually floats in the air so that you don't come across any sort of vibration. And you can actually see this glass tip, which is actually what we call as a perfusion pencil. It can be shown over here and also here. So what it's doing is that it's actually coming closer. You have your cell plated on a dish with transfected receptors, which goes on in the center here. And using the glass holder, what's actually it's doing is it's actually, coming close and patching on to a single cell membrane and making a small patch. And by making this small patch, it actually, is making it completely isolated from rest of the cell surface, from rest of the cell. And in this way, you can record. So the amount of receptors and ion channels you get in touch with this patch, you can actually record, the influx and outflux of the ion channels and, different proteins, and study the activity of the G protein receptors. And this is actually connected to a feedback resistor and the amplifier, which is, modulated. And then, you can use your different agonists and drugs to study the effect of different, peptides. So yeah, that's what I actually like to do. I'm actually an electrophysiologist, and that's what I've been doing over these years. So yeah, to study these transfected mu-opioid receptor, we transfected them in a mouse pituitary tumor cell line, the ATD20 cell line. And they had native expression of somatostatin receptors and potassium channels and different versions of the mu-opioid receptors like we discussed before. They were transfected in there. I won't go too much into this because I can explain that in the next slide. The whole take-home message was that you actually need a recording protocol to study whatever type of protein channels or ion channels you have, because the different types of ion channels inside your cell membrane or your cell body, they operate at small, tiny voltages. So for example, the potassium channel that I was studying in this, it was fully functional at minus 120 millivolts. So using your amplifier and your patch clamping condition, we held every single cell at minus 60 millivolts, and then it was pulsed to minus 120 millivolts. And you can see over here that the black line over here is just with the potassium, 20 millimolar potassium buffer. But when I applied Met-Enkephalin, the amount of current injected was like double the original black current. And then, based on, you take the difference between these two, and then, you plot your graph. So the rest of the whole slide I will show you will be actually the difference between exactly, the difference between the red line and the black line. Yeah, so like I said, the whole idea was to actually study these mutants under patch clamping condition to look for opioid receptor desensitization pathways. So I'll just go through this very quickly. Using the mu-opioid receptor wild type without any C-terminal tail mutation, when I patched onto the cell and I applied, M is for Met-Enkephalin. So when I applied opioid Met-Enkephalin, 2 pulses, and then I applied it at high dose at 10 micromolar for 5 minutes. And you can see how the signal is going down within this 5 minutes, even though the application is continuous, which actually shows the receptor to be desensitizing within 5 minutes. And then using the same pulse I applied over here, you can see how the response has gone down as compared to what it was before. So this is actually called receptor desensitization on the surface. That how after a strong exposure, the receptor gets desensitized, but then after washing it over a period of time, the response keeps coming back and back to the original baseline where it was. So this actually got very interesting in the 11-STA mutant, which was a mutant where all the 11 C-terminal tail mutations were mutated. They were all mutated to alanine. And you can see how over here, the signal doesn't drop on applying Met-Enkephalin. It remains constant. And the signal over here is pretty much similar to what you see before, continuously throughout. So somehow the C-terminal tail mutation is completely abolished, the desensitization which you see in the wild type. So there you go. In the wild type mutant, and there was significant desensitization, but 11-STA still produced 100% response, no desensitization at all. But the 6-STA and the 3-STA, which we tested, they still desensitized. But in another set of experiment, when we applied morphine, we got a bit of a different set of results. So keeping in mind, we repeated the same experiment again, what we did for Met-Enkephalin. Applied Met-Enkephalin here, and then applied morphine here for 5 minutes. You still see desensitization, and you still after washing out. The only difference is that it's not recovering back to the original baseline, what you saw previously. And with 11-STA, it is still desensitizing with morphine, and it's not recovering back to the original baseline that was seen before. So this was very interesting because this actually gave us an idea that there is something different between morphine and Met-Enkephalin, maybe two different set of signaling pathways. So after Met-Enkephalin, you see a reversal back, but with morphine, there is a huge amount of desensitized receptor still left, which doesn't recover back. So what is actually happening here? We thought, okay, let's just try an intracellular blocker like PKC to see if we can block the remaining part of the desensitized receptor. So this is with Met-Enkephalin. Again, in the wild type receptor, you still see receptor desensitizing and the response is small. With the 11-STA, no effect at all. It still doesn't desensitize at all. But with morphine, when we applied it on the wild type receptor and applied a PKC blocker, which is Calphostin C, if you remember, because previously the desensitization was pretty much strong around here. Now there is a lot of a recovery seen initially, and the 11-STA, it recovered back completely. So somehow the intracellular PKC is regulating the morphine-induced receptor desensitization. So when you block PKC, it abolishes desensitization with morphine. I'll just go through it because I am a bit running late on time. So yeah, this cartoon will sort of help. So whenever you apply Met-Enkephalin on the membrane receptor, it activates G protein receptor kinase and phosphorylates the C-terminal tail, because when you mutate them, it leads to receptor desensitization and it leads to receptor desensitized receptor. But in terms of morphine, because we had the 11 C-terminal, 11 serine threonine residues mutated to alanine, but when you mutated them, it was still desensitizing. Why? Because another intracellular molecule like PKC was coming forward and somehow regulating the receptor. Okay, so when you're actually doing research, that's how you go forward. You actually start your experiments in an in vitro small model, and then you take it to the higher models. So the next step was to study this in, a mouse model. So because we knew that the 11-STA, the 11 mutated residues, they actually don't do any desensitization of the receptor. So we used, knock-in mouse model. And like if you remember in the initial slide where I mentioned to you that the locus coeruleus region of the brain is the region where lots of the opioid receptors are present. So our lab built in an 11-STA knock-in mouse. And the idea was to take the brain region, which is this tiny, like it's the size of your fingernail over here, and then take the slice region, do a slice cutting, and take the slice region which has the locus coeruleus region. So this is actually like around a 300 micrometer thick slice, and you can only get one slice out of a whole mouse, on which you can put your recording electrode on the specified region, because it's so hard to identify, and the cells can die very easily. So just use a recording pipette and then again record the desensitization via Gurk channels. So yeah, again, taking the story from the cell lines, building a knock-in mouse with 11-STA. And this is a wild type cell line with just showing the expression of the mu-opioid receptors. So in a wild type mouse, again, repeating the same electrophysiological approach that we use for the cell lines. When we applied Met-Enkephalin at a higher dose, you can see how the current is declining, means that the receptor desensitizes and it recovers back to the surface again. But in an 11-STA knock-in mouse, you can still see that the amount of desensitization, it's still there a bit, but there is a huge amount of differences compared to what we see the wild type, that somehow the desensitization has been significantly reduced in the 11-STA knock-in mouse. And this study was actually published in 2019. And not only that, apart from electrophysiology, we did some animal behaviors, the pain behaviors, to actually study the effect of different opioids. So I'll just go through some basic points here. So since opioids are good analgesics, and when we talk about tolerance or addiction, just focus on this black bar graph. So this is a wild type mouse, and the idea was to study it under a pain model. So this is basically, just putting a mouse paw on a hot plate, and just to look at the time it took for it to withdraw its paw. And then, injecting the mouse with an opioid like morphine or fentanyl, or and then, just using the different animal models, how much time it took. So for example, when you give fentanyl to a mouse, wild type mouse, to see how much, and you inject its paw, the amount of time it's actually taking. So, in 15 minutes, the latency, the amount of time it took for him was like this much, but then, it starts to drop down, means like, it's just becoming more tolerant to that dose. But the 11-STA and the 10-STA, there is still an extreme amount of analgesia found. So the mouse can actually expect a lot amount of pain as compared, the 11-STA mouse can expect a lot amount of pain as compared to the wild type mouse. The same goes for morphine. The amount of analgesia has been remarkably increased in the 10-STA and the 11-STA mouse as compared to the wild type mouse, showing that somehow the 11-STA, the phosphorylated sites are basically really important in mediating the opioid analgesia and reducing side effects. So, hopefully I can finish this up in the next 5 minutes. So yeah, we actually understood the story, we actually understood the mechanism, and we understood that how the C-terminal tail is actually important in mediating receptor desensitization and mediating, tolerance and addiction. But how can we actually use this approach to design a safer opioid? And somehow, can we use these parameters by interfering in these regions to design a drug or to identify a drug which can help us identify safer drugs? Another theory about the opioid receptors is, like I said initially, that when any different opioids can activate different signaling pathways. And that phenomena is actually called as opioid bias, preferring one pathway over another pathway. And pharmacologists have been designing different drugs, based on this theory, so that, just so that, you can have a safer opioid. So our lab in 2019 published this paper at PNAS, in which we identified a novel marine fungal peptide bioverse, and we studied its G protein activity. It was found to be an opioid peptide with strong G protein activity using cell lines. And then, this green line is basically, the blue line is basically Bioverse showing its concentration response curve as compared to morphine, and Met-Enkephalin showing how much its activity has been reduced, its efficacy is a bit reduced as compared to Met-Enkephalin. And then, studying its arrestin binding pathway and its internalization pathway. But these parameters were found to be really important when we compared them in this bias signaling profile, showing that it actually has a strong G protein binding trend as compared to internalization trend. So I'll just go this one last slide, and just the final message was that by applying Bioverse using the same desensitization profile, Bioverse was meant to be signaling majorly through protein kinase C as compared to Met-Enkephalin or endomorphine. I'll just skip this. And final take-home message was that by using Bioverse, a modified version of Bioverse, which was Bylactorphin, and giving it orally to a mouse model, it definitely improved the amount of analgesic profile and significantly reduced the safety profile, just by focusing on the approach that if you focus more towards the G protein signaling as compared to the internalization signaling. So finally, the concluding remarks are that the C-terminal phosphorylation tail is the first step leading towards mu-opioid receptor desensitization. Different opioids signal through different signaling pathways. Met-Enkephalin signals through GRK and morphine through PKC pathway. Phosphorylation deficient mice shows enhanced analgesia and reduced tolerance. And upon increasing G protein activity, opioids tend to signal majorly through the PKC pathway, leading to receptor desensitization. And finally, Bioverse was a naturally discovered biased opioid agonist with strong G protein activity and shows significant analgesia in mouse models. And finally, I would like to acknowledge the lab members with whom I worked with for over 7 years, especially my boss, Professor Mac Christie, and all my team members over here, and also to my collaborators in Germany. And now I actually acknowledge you, and I can, if I have time for questions, I'm happy to answer your questions as well.
Hello, my name is Ruhma Tahir. You presented very well. My question was that in one slide you explained that even when a person is not taking opioids, he still is having some signals via the receptors that a person is he is exposed to some kinds of opioids and a non-adrenaline is rushing in his body. So how is that even possible that even when the drug is not, that the person is not in taking the drug, but there is the response of the drug or the signal is still roaming around the body and adrenaline is produced? Hello, can you hear me?
Yeah, I can hear you. I'm just going to take out a slide. Can you still see my slide?
Yes, yes, I can see your slide.
Okay, so the whole point is when we talked about tolerance and addiction, what actually happens throughout, like I already mentioned, is the brain, the nucleus accumbens, when it's actually triggering back via, the prefrontal cortex back into the locus coeruleus region. When if you're taking an opioid under a low dose, your body can tolerate, for example, morphine, the normal dose, which the doctors prefer to give is like around 30 milligram or 20 milligram. But under a serious chronic condition, when the doctors, they actually expose the patient to morphine for like weeks, for months, that's actually the problem, that, your reward center, and because your membrane receptors in the brain, they become seriously activated and desensitized. And because of the amount of nucleus accumbens and about amount of the noradrenaline that has been activated in the meantime, that keeps on craving for more and more. It actually sort of makes it, because it actually needs more and more reward system to actually, overcome the amount of, so it's actually a feedback loop that craves and craves. And this period actually keeps on happening for about, let's say, 5 days to 7 days in a human being, until, the receptor machinery, it gets back into a normal phase. So in the slides where I showed you how the receptor desensitization took place and how much time it actually takes for the receptor to recover back, it actually takes that time back for the body to function normally in that period of time.
Thank you so much.
Sir,
Walaikum salam.
so my name is Kutumban and I have a question, a general question. It's about like a physiological conditions. One of my relatives is hit by a car and suffered from an accident. His pelvic bone was cracked that time. And now even it is healed, but now he feel pains even after healing and even after recovering from that accident. And we have concerned with some neurophysiologists and doctors that it's about like maybe his nerve are damaged or maybe his neurons are damaged. So they said the neurons or nerves are completely okay. Then why this condition occur when it's get cold? He feels pains in that pelvic area where he got hit by a car or where his bones are damaged.
Yeah, I mean, that's basically the condition of chronic pain we have studied. So what happens is, when your your neuronal body, the nervous system, especially, in the dorsal horn, especially, so these nerves, they are like very fragile. But what happens is when some sort of, unfortunate reasons, whenever there is an injury, it's like very hard for them. So, I mean, they do sometimes, if they do even recover back, but somehow, their signals, their nerve. So for example, their nerve recovery can take some sort of a loop region in which an alternate pathway is triggered. And that's actually, another, like a class of receptors that still keeps on signaling back to the brain. It still keeps on firing back to the brain. So yes, you're right. The pain itself isn't there, but the response signal, the brain, the somatosensory signal, still keeps on firing to the brain, and telling actually, that so it's basically, it happens as a plastic change to the nervous system, to the spinal cord, that still keeps on triggering back to the brain that, and there is a damage there. And it's very unfortunate that happens. Look, I mean, I'm not a physician myself. I can just recommend you to go to a good neurophysician doctor if you have around there, or maybe like definitely, I think in Lahore, just tell them the complete story, or like, get a proper scanning of the damaged area. it definitely needs to be treated as a chronic pain condition.
Okay, sir. Thank you.
Assalamu alaikum, sir.
Walaikum salam.
Sir, my name is Sharias, but and I want to ask a question, sir. Whether the opioids itself insensitize the G protein linked receptors, or it releases dopamine and then it insensitizes it, and why the effect is reversed when it occurs naturally and not reversed for even weeks and months when we take an opioid.
No, that's a good question. So opioids itself, they can trigger the G protein, the mu opioid receptor. What actually, because in the brain there are different regions which gets activated. So you have at one part you have the locus coeruleus system that gets activated, and you have specifically the mu opioid receptor targeting and its activation. But somehow opioids also act on dopamine class of receptors as well. So the opioid receptor signaling is pretty much specific to the mu opioid receptors, but it's also acts on the dopamine receptors and onto the noradrenaline receptor as well. In regards to your second question, look, I mean, the phenomena of receptor desensitization is very unique. The whole idea is in most of the G class, it's actually called a class A G protein coupled receptor system. In most of these systems, I mean, there is actually a natural way, and then there is a synthetic way. A natural way, all these opioids, you have got them naturally produced in your body. You've got naturally produced endorphins, enkephalins, which are actually naturally produced in your body. There is a natural way how your body has been designed to keep you active. And for some unfortunate reasons, if some sort of unnatural thing happens to your brain, even the reward system or the addiction system can be triggered. But by using a synthetic way, like by orally taking a high dose of, so it won't actually happen under a low dose. Like the animal models which we have studied here. If you give them under a minimal dose, you won't see much of a craving or much of an addiction. But all these animal models, they have been given under a very high dose. For example, morphine, it's like, they have been injected at 20 milligram to 30 milligram, which is already, it's, over the top limit for dose for, and fentanyl itself is like strongly potent, but it's a synthetic opioid. So that's the problem which we have been trying to address, that how fentanyl, which was actually, it was identified to be a good analgesic and as a potent analgesic, why its side effect profile was so strong, because it was targeting all these other important pathways within the opioid receptor system. So by that's what actually happened in the US. So our approach always was to, if you want to design an opioid, just make sure that its G protein activity is not that strong. See over here, like Met-Enkephalin, this is actually its G protein activity, how much, it's like around 80% as compared to the control. And if you don't have that control, it's pretty much will reach around 100%. But in order to design an opioid, you have to make sure that its efficacy, we call this as its maximal efficacy at a higher dose, that it should be like pretty much 50% or something, so that it doesn't hit that ceiling, so that it can cause an overdose. Let me just pull out another slide if I have. I don't think so. I have it here. So there is another opioid called buprenorphine, which has like around 20 to 30% of its activity, of its efficacy. So the whole point is, the stronger the efficacy it will have, the more signaling pathways it will start to activate. And the most important signaling pathways which people have been working on is the receptor internalization pathway. You need to have a strong dose of an opioid for the cell membrane receptors to start internalizing into the cell. And that's why, it takes a lot of time from the cytoplasm for it to reach to the cell surface. So the whole point was to reduce the efficacy profile, to reduce the sealing profile, so that it can have a small G protein activity as compared to, small G protein activity as compared to the other opioids. And that's how, pharmacologically, we have been able to design a safer opioid. So that was basically the take-home message.
Thank you, sir.
Assalamu alaikum, sir.
Walaikum salam.
Sir, I want to ask a question that when you used morphine on 11 STA, so there was a little bit, there was desensitizing, the signal drop. So why is that in case of morphine, but that doesn't happen in case of Met-Enkephalin? And same, and further it doesn't recover back also. So what's the difference between morphine and Met-Enkephalin?
So are you talking about this data here?
Yes, sir. So why morphine behaves differently?
Yeah.
And it also doesn't recover back in 11 STA.
Yeah, so that was actually the convincing experiment that was shown in the next slide. So I can go through this again. So the whole idea was, let me just pick this one up. The whole idea is that different opioids, they target different signaling pathways. So Met-Enkephalin, it targets signaling via beta-arrestin and G protein receptor kinase. So, when you apply Met-Enkephalin, it pretty much phosphorylates all the serine residues.
Excuse me, sir. Can you explain this again? The last previous slide. Can you explain it again?
Which slide are you talking about?
Actually, the previous one before this one. Actually, I think so. It's
okay.
down.
Okay. Yeah. Okay. This one. Yeah. Or no, I get it. The cartoon. This one. Okay. Okay. I'll come back to this, but I think this is more important to understand. So when we actually started, so using a phosphosite specific antibody. So if you look at these colored dots here, all these are actually site specific antibodies. And over here is the time frame. So, after 20 seconds, 40 seconds, this is actually all Western blotting done after treatment with Met-Enkephalin and after treatment with morphine. So with Met-Enkephalin, it actually is phosphorylating pretty much all the sites, but morphine isn't. That means that, with Met-Enkephalin, you're still getting a huge amount of phosphorylation of all the serine residues. But with morphine, you're not phosphorylating all the sites. So that's why when we removed all the 11 STA mutants, the desensitization was gone with Met-Enkephalin. But with morphine, because it's not phosphorylating all the sites, another intracellular pathway, which is PKC. So in morphine, another intracellular pathway, PKC, is regulating the receptor desensitization. And then that was actually checked with patch clamping, in which when we applied Calphostin C on top of the cell with Met-Enkephalin, you can still see that the wild type still desensitizes and the 11 STA doesn't desensitize. So pretty much what we saw before. But morphine, on applying PKC blocker Calphostin C, the desensitization is pretty much gone in the wild type, and it's it's reversed. So the difference between morphine and Met-Enkephalin is the difference in the signaling pathways that are actually getting activated. So that's the reason.
I'm sorry to interrupt. I know all participants have a lot of questions, but we are running out of time. So now just last question.
So I have a
Yeah,
question.
sir.
So,
Yeah, sure. Please go.
so I have a question. Morphine or Vicodin or heroin, all of these actually lead to like overdose, addiction, and death. So why do you prefer these kinds of medication for pain? And why do everybody else actually go for these medications for pain relieving? And because obviously it's all going on your head. So it it actually gets you high at one point and you get addicted to it. So why do we are actually recommended to use it? Why doctor actually prescribe it? That's my question.
Yeah, that's a good question. Look, I mean, the thing is, opioids, they are very good analgesics. I mean, if you're looking for patients at chronic pains, especially, in a country like Pakistan, where there is not much of a pharmacological research and where the pharmacological drug imports is also very much, you don't get much of a pharmacological import of good drugs or expensive
Yes.
drugs. That's why, the doctors over there, they are pretty much stuck with the amount of, drugs which they have. The other thing is what doctors actually recommend that along with morphine, they give you an antagonist drug of the mu opioid receptor. Like normally, sometimes they will recommend naltrexone or naloxone. So the idea is that it actually covers the side effect part. So, it actually, along with, hitting the opioid receptor, it actually blocks the opioid receptor activity as well. Look, there is novel research. So when I joined University of Wollongong over here, I've like focused from the mu opioid receptor to GABA B receptors, which is also like, opioids along with targeting the mu opioid receptor, they also target the GABA B receptors. And the whole idea is, to look for other proteins and look for other medications that can target other receptors apart from the mu receptors.
So sir, you say morphine or opioid, they can actually cure chronic pain as well as nerve pain. What do you say on that?
No, it doesn't. It doesn't actually cures it completely. I mean, the thing is, it is way much more stronger in its efficacy at the moment as compared to other drugs in the market. Oxycodone or heroin or like fentanyl, they are very, very good analgesic drugs. They are actually very good analgesic drugs to treat chronic pain. Like a person with a serious chronic injury, they are very effective analgesic drugs, no doubt, because the whole point is that, I mean, I can go into a bit of a complication here. The whole point is with about, you just need 20% of the receptor to be active to give you a strong, strong analgesic response. That's the beauty. That's how strong the mu opioid receptor rewards analgesic system is. So even by giving a small amount of dose of fentanyl or morphine, you can actually create a strong analgesic response. That's why the system is like,
We actually won't be 20% open when we're gonna get induce
yeah,
more amount of.
exactly,
Okay.
exactly.
So one last question: is
Yep.
chronic pain and nerve pain related?
Look, any nerve pain, any nerve pain that keeps on persisting for more than 6 weeks, it becomes chronic. So any sort of a nerve or a nerve tissue injury that keeps on persisting even after you stop medication, and it keeps on persisting even after the tissue heals, it actually is called a chronic condition. Because like one of the students actually asked the question that it still is, you still have a persistent firing of the response. Look, what pain is, or whatever your tissue or whatever your brain feels, is basically, the firing of the nerve signals that keeps on triggering to the brain. If your brain, if, somehow some of these ion channels, for example, sodium channel, there was an interesting case from Pakistan that was reported that he had sodium channels mutation somehow in his body, naturally occurring sodium channel mutation. And he just couldn't feel any pain, because sodium channels are the first ion channels along with potassium channels that actually, trigger the action potential, trigger the signal from the hand to the brain. So if your body can't for the signal, your brain won't even feel anything. But if that signal is somehow, disturbed or destroyed, or it's mutated in a way that it keeps on firing, your body will still keep on feeling pain even after, for some
Do these
unfortunate reason.
kind of channels depend upon actually the state of neurons and the muscle cells?
Well, I mean, your sodium channels are pretty much present everywhere. They're pretty much present in your neuronal cell, on your muscle cell as well. They pretty much depend on, the threshold membrane potential of the cell. So because there are all of these channels, they are voltage operated. So if you go more into the biophysics, whenever, there is a signaling response, the threshold level, the biophysical membrane voltage of these channels, they get, a bit depolarized. And that's how, the biophysics mechanism happens. They start firing and transfer the signal forward.
Thank you, sir. Thank you so much.
All right. Thank you, Dr. Arsalan. Thank you, everyone who participated and asked questions. I think that's enough for the questions. Now I would like to call Dr. Mohammad Sohail Afzal for concluding remarks.
Assalamu alaikum, Dr. Arsalan.
Wa alaikum assalam.
I would like to thank you for your time for today's session, and it is nice to see you after a long time. And
thank you very much.
It is a pleasure to hear you on the subject. I would like to acknowledge your hard work and contribution toward the subject over the period of 15 years. And it was a great experience having you on board today with us. It was excellent to know about the role of G protein receptor under the activity of opioids. I hope our student learned a lot from your lecture today, and I hope this might help them in the planning of their research in future as well. I would like to thank organizers and participants from the Department of Life Sciences. And I hope such activities will continue in the future, inshallah. And Dr. Aslan, we are looking forward to collaborate with you in the future as well. And thank you once again for your time, precious time for the today's session. Thank you.
Great. Thank you very much, Dr. Sohail. It was really nice to hear your voice again. And thank you very much for the organizers to invite me on board. I hope I have, answered maybe not all of your questions, but maybe up to a limit that you might be able to understand this area along with me, because, I'm still learning. Learning never stops, and it's an ongoing process. With every question I get, it encourages you more to learn a lot more. So I'm very much excited to be here, and I would definitely look forward to interact with anyone from your lab or from your university or from your department for the future as well. So thank you very much.
Thank you.